My laboratory is focused on the physiology of disease-transmitting mosquitoes.
Emo Hansen, an associate professor at New Mexico State University's biology department, has received a $1.46 million grant. The grant is funded by the National Institution of Health and will allow Mr. Hansen to study amino acid transport in mosquitoes for the next 4 years.
Mosquitoes have a very special diet. They feed on blood, as you know, and blood is a very protein-rich diet. So these proteins get digested into amino acids, and then the amino acids are made into mosquito proteins and also into energy.
The goal? To lower the mosquito population in our community.
The idea behind the research is to find new inhibitors to inhibit the amino acid transports and inhibit mosquito reproduction and egg production.
But that's not the only attempt at reducing mosquito population.
Right now we have an ongoing project where we try a new technique to control mosquito populations that does not involve pesticides. This technique is called sterile insect technique. And the idea is that you release sterile males in the wild, and these sterile males mate with the females, and then the females lay eggs that never hatch.
The male mosquitoes become sterile after being exposed to low levels of radiation.
We're talking a very limited amount of X-rays are actually going into this mosquito. It's snapping in the gametes, so sperm cells in males. If we can release a lot of male mosquitoes that are sterile, they mate with females, and we can dwindle the populations down to a very, very limited level and not necessarily eradicate them, but suppress them, we can prevent disease. If you're here in the North Americas and South America, it would be dengue fever, chikungunya, Zika, that sort of thing. So if we can eliminate those, then we can ultimately make an effective vaccine without ever having to use humans.
With Aggie Vision, I'm Matthew Nieto.